Good afternoon, everyone. So we'll start with the technical session. I'm pleased to welcome you all to this technical seminar we have arranged on behalf of Karnataka Poultry Farmers and Breeders Association and Karnataka Veterinary and Fisheries Science University. From last 2 years, we are arranging these technical seminars. This is a collaborative effort by the association and the university. Today, we have with us Dr. Tosh, who is a principal scientist in Bhopal lab and an expert on AI as recognized by World Organization for Animal Health. So I request Dr. Tosh to come and occupy the seat, please. Please take your seat. I request Dean sir to come and occupy the seat, please. Okay. And please you take the seat. Yes, sir. Both of you can take the seat. Okay. I request Dr. Islur to come and occupy the seat because it is Dr. Islur who is instrumental in bringing Dr. Tosh to this meeting. I request our Association Secretary, Prasanna sir, to come to the dais. Please occupy the seat, sir. Okay. With this, I request Dr. Islur to give a brief introduction about the speaker. Once he gives the introduction, then all of us will go back and sit in there, and Dr. Tosh will do his presentation. I think presentation would run into about 45 minutes to 1 hour, followed by a discussion. We expect more of a discussion because majority of us know what is AI and majority of us have experienced AI. So all of us know. So it's I would suggest that we ask more questions and interact with him so that you can know more and more about it, which we do not know, or whatever the latest developments in this field that has happened. This is an opportunity for all of us to know it through Dr. Tosh. I request Dr. Islur to give a brief introduction about Dr. Tosh. Thank you.
ಡಾಕ್ಟರ್ ಹರ್ಷ ಅವ್ರ ಹೈಟ್ ಇನ್ನೊಂದು ಟಾಂಬ ಟಾಲ್ ಆಗಿದ್ದಾರೆ, ಅದಕ್ಕೆ ಕೆಳಗಡೆ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಧನ್ಯವಾದಗಳು ಆತ್ಮೀಯ ಮಿತ್ರರಾದಂಥ ಡಾಕ್ಟರ್ ಹರ್ಷಕುಮಾರ್ ಶೆಟ್ಟಿ ಅವರಿಗೆ. ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. ಗುಡ್ ಆಫ್ಟರ್ನೂನ್ ಟು ಯು ಆಲ್. As Dr. Harsha said, today we are here in connection with a technical seminar followed by interactive session, as he had rightly put it forth, on one of the very, very important topics, avian influenza, with respect to its current status and what are all the current control strategies at the global level as well as the national level as well as the regional level. And no exaggeration, I'm so pleased to have one of my best friends, Dr. Tosh, as the speaker. This is not vote of thanks, Tosh. Thank you so much initially itself for having obliged all the way coming down to Bangalore from NIHSAD. ತಮಗೆಲ್ಲರಿಗೂ ಗೊತ್ತಿದೆ. He's from ICAR, Indian Council of Agriculture Research. NIHSAD ಮುಂಚೆ ಅದಕ್ಕೆ HSAD ಅಂತ ಹೇಳ್ತಾ ಇದ್ರು. That was known as High Security Animal Disease Diagnostic Laboratory. Now it is actually represented as NIHSAD, National Institute of High Security Animal Diseases. And we are so pleased and feel, especially myself, feel so proud of one of my best friends, my batchmates at Mukteshwar. 35 years ago, we had a chance to pursue our post-graduation in veterinary virology itself from 1989 through 1991. 20 साल बाद नहीं, 35 साल बाद. So for the decades they themselves are actually passing. And you know about avian influenza, how important the disease is, and no need to emphasize the global One Health importance the avian influenza is actually gaining. Globally we speak of One Health, wherein It is not just the animal health component, not just the human health component, but also the environmental health component also pitches in there. It is the interaction between these three important components that actually leads to occurrence or non-occurrence of a disease. So also with respect to this avian influenza. We all know how this avian influenza virus is changing its genetic profile or the phenotypic profiles that has actually resulted in so much of concern. ನಮಗೆಲ್ಲರಿಗೂ ಕೂಡ ಗೊತ್ತಾಗಿದೆ ಇಷ್ಟು ವರ್ಷದಲ್ಲಿ ಇಟ್ ವಾಸ್ ಆಕ್ಚುಲಿ ಜಂಪಿಂಗ್ ಫ್ರಮ್ poultry waterfowls to aquatic waterfowls to the domestic poultry, but what has happened very recently in the United States, dairy animals have picked up the infection. Why go elsewhere in our own country? In Madhya Pradesh, isn't it, Tosh, the first episode of a jumping of virus to 3 domestic cats just in the month of January 2025 itself. Other than the leopards, felines, big cats, the disease has been reported and they have succumbed to the disease. In this context, the One Health-based strategy is really going to play a major role, not just elsewhere, but even in our own country. So, in that context, having this KPFBA and KVAFSU joint collaborative efforts have resulted in having this very, very important topic, technical seminar on avian influenza. What is its current status? And then what are the implementable strategies or what is already there in the paper, and then global health, and we need to put this into action at the local regions. So in this context, Tosh is here, and you know Tosh in fact is basically a veterinarian like most of us here, and then he did his BBSc from Odisha Veterinary College way back in 1989. He completed that, and then in 1989 he joined IVRI Mukteshwar, where myself and Tosh, both of us were together to pursue our degree in veterinary virology. And then after his masters, he did masters on foot and mouth disease, and then he continued to do his PhD program in foot and mouth disease at ICAR-NIHSAD itself. But in 1994, he in fact joined for the ARS system. ICAR will be an agricultural research scientist position. He joined there, subsequently completed his PhD, and he is having so much of expertise in foot and mouth disease. Then he did a lot of work on PPR virus, and then now he is known as man of avian influenza in India. No exaggeration, Tosh. So his basic association with respect to avian influenza or other viruses is the host-pathogen interaction. The host is also playing equally important role as far as occurrence of disease. Then the molecular epidemiological aspects. Then development of variety of diagnostics as well as the variety of vaccines. They're all his expertise, which he has been extending not just to the India, not just to our country, even to the neighboring countries. As a OR designated expert on avian influenza in the country is concerned. So no need to emphasize that he is having many number of publications, patents, and then workshops. He has been the resource person, not just locally but elsewhere. So with this brief introduction, thank you so much, Tosh, once again, and now it is over to you, Tosh. Yeah, thank you.
Thank you. Namaskaram. Good afternoon to all of you. And at the outset, I would like to thank the organizers for this opportunity and the support. NIHSAD is not only working on influenza, it is working on the exotic and emerging animal diseases, including zoonosis, like ASF, African swine fever, and lumpy skin disease, besides avian influenza. And this NIHSAD houses a OR reference laboratory, and also this is also acts as a national reference laboratory for avian influenza since 2009. And as you know, this is actually a BSL-3 laboratory established in the animal sector to cater the needs of any exotic diseases entering into these territories for its preparedness and control measures. So, how to move? So this will be my outline of presentation: situation updates, India and the global situations, and also the surveillance systems in place for control and containment of this disease, in poultry, and then the control measures what is already in globally available and India following. So this will be in brief. You can interact me in between or in each slide. Each slide you can interact. No issue for me.
So can I, if you don't mind?
So, as you know, this is a transboundary animal disease. As you know, this transboundary means it is highly infectious and spreads very rapidly. And if it is not attended at proper time, then it becomes endemic. And also some diseases also having economic impact, and also it affects also the international trade. So it is a highly and viral disease affecting domestic poultry, mostly the meat producing birds like chickens, turkeys, guinea fowls, and quails, and also wild birds. More than 100 species of birds have been infected since last 50 years of research and surveillance, starting from 1970s. It has been found that wild birds, specifically the aquatic habitat birds like wild ducks and geese or gulls or the shore birds, these acts as the reservoir species. But now these birds are also dying. The HPI virus is jumping from poultry into the wild birds, and they are also succumb to this disease, highly pathogenic. And also mammals, if you see, it is sea mammals and the land-based sea mammals like seals, and the land mammals like you have seen in recent past, the domestic cattle, sheep and goats, they are also susceptible to this disease. And also humans, sporadic human infections have been reported at least in 25 countries, and more than 2000 infections has been reported from different subtypes. Like you have seen that there are H5 subtypes, H7 subtypes, H9 subtypes. Though it is a low pathogen, still it infects humans, and there is of course self-limiting, but still there is death due to LPI H9N2. And why it matters, like it, as you know, it affects livelihood of farmers, in specifically in the low and middle income countries. Once this disease occurs, the control measure is in India, government of India is the culling policy, and then sanitation. Once it is called and forced culling and compensation, the restocking is delayed for at least 60 to 90 days minimum. So during this period, he or she cannot restock any poultry. So it affects a lot in the livelihood of farmers, in specifically in the low and middle income countries, including India. And then international trade. If you have the disease in your poultry, then your poultry you cannot export to any country. So, and there's again it is affecting biodiversity. See, because it is again lot of wild birds during last 4 or 5 years it is affecting and dying. So it is affecting biodiversity and One Health. This is one of the real issues of One Health implications. It is affecting human beings, it is affecting the poultry, it is affecting the animals and also the marine animals. So it is One Health implications. And in view of that, the government of India developed an action plan in 2005 to deal with this disease in India, how to control this, and this was circulated to all the states and territories in 2005. Then subsequently, based on the experience gained from the outbreaks, it was revised from time to time. Now it is the revised version 2021. That is the last version. You can intervene in between and ask questions. Of course, these are the introduction parts. And as I discussed, wild arthropods like dogs and shorebirds, they act as reservoir species. They don't say any clinical symptoms. They just carry the virus, shed the virus into the environment, then fresh birds are infected. So it goes on, the cycle goes on, and these viruses are shed through the respiratory or the cloacal sheddings, and it contaminate the environment, and any environmental contamination can spread the disease to the naive poultry populations. Like direct bird to bird contact can infect the healthy birds, and also spread by through contaminated surface material, specifically those who are feeding or watering the poultry in the farms, if they are having the poultry in their backyards, they can carry the virus to the farms through their shoes or their dress materials, and through any inactivated materials also they can carry. And migratory waterfowls also they act as a carrier, and also the local migratory birds, like they are not the real long distance migratory birds. Local migratory birds, they also play a critical role in transmission of disease from one place, from infected premises to the healthy premises. And these are other also where the smuggled poultry, if you don't know the source of the poultry, if you are bringing some poultry and they are having the virus, they can also spread the disease to your poultry farms. And also the people also play a role in the transmission of the disease. As I told, if somebody is working, those who are working in the farms, they can spread the disease from infected farms to the healthy farms also. And these are the ways how the virus can be spread. This virus can survive in the manures, and they can be transmitted through the equipments, and by clothings and by shoes or hands. And human infections, mostly globally, it is through the directly from the infected poultry or those who are in the processing of the poultry there. And of course, luckily there is no direct human to human transmission is there, not there. Very limited clusters of human transmission within the family is only reported in very few countries, including Vietnam and other countries. But there is no direct human to human transmission. Only those who are in close proximity or those who are engaged with poultry processing or poultry farming, like the waterers or the laborers, they are only highly susceptible to this disease. So about this virus, there are 4 types of influenza viruses. Influenza A virus is having, it infects multiple species, including humans and poultry. It can jump to pigs and the horses. So then different classes of there. And based on this, there are 16 HA subtypes. This virus having hemagglutinin and neuraminidase. Based on this, the virus proteins, the virus is classified into different subtypes like H5N1, H5N6, H9N2, LPI. So there are known combination of viruses, though there are 144 combinations, but still 133 combinations have been isolated from the wild birds. And you must have heard about low pathogenic, and there is a high pathogenic. In low pathogenic, example is in our country, it is H9N2, where in laboratory settings it does not kill the birds, but in farm settings it kills the birds because of the other organisms like E. coli or Salmonella or other viruses like ILT or IAB. But as such, LPI virus H9N2 in laboratory condition does not kill any birds. As compared to highly pathogenic viruses, they kill in the laboratory condition within 48 hours, 100% mortality. And this behavior of this virus in the field is different. Does not kill within 48 hours. And this is only related to the chickens and the poultry, low pathogenic or high pathogenic. And these subtypes, specifically the H5 and H7 subtypes, has mutated to become highly pathogenic avian influenza. In India, only we have reported H5N1 and H5N8, but S7 subtypes in other countries like in China, they have S7N9 subtypes, and in other countries like USA and Europe, they have S7 subtypes with different N combinations which are highly pathogenic. But in India, we have only H5N1 and H5N8, which has been reported from poultry and also other birds like the wild birds or the peri-domestic birds. Lot of mortalities in crows in India. And these 2 viruses, since they are mutable to high pathogenic and also of human concern, they are reportable diseases. This has to be reported by the Department of Animal Husbandry. and also to the World Organization for Animal Health. So these are some of the recent outbreaks to highlight before you, since there is poultry outbreaks, wild birds, and this year we have still spillover to wild mammals and also companion animals in pets. In December, 15th December, mortalities were reported in cranes, specifically the demoiselle cranes. They are long distance migratory birds, and in Rajasthan, and we received dead birds. NIHSAD received dead birds, and they were tested positive for influenza A and H5N1 by real time and the virus isolation. These are the standard methods by the laboratories which are tested and isolated, and the whether it is highly pathogenic or not, we sequence these viruses, and based on the sequence analysis we got these are the highly pathogenic. In highly pathogenic and low pathogenic, the hemagglutinin have multiple basic amino acids where it can cause systemic infections, so that's why they are highly pathogenic. But for low pathogenic avian influenza viruses, they are very restricted to the guts and the respiratory tract. They are not systemic, so they don't kill the virus specifically. They have self limiting disease, only there is production losses, and also like in the form of egg production or in the meat, but they don't kill the virus in the laboratory. But in farm conditions there is mortality maybe 2 to 3%, or it may increase depending on the age of the birds or the different immune status of the birds. Then subsequently, after this Rajasthan outbreaks, we have different, at least 10 to 11 states reported a high pathogenic avian influenza in poultry. It's Maharashtra, Chhattisgarh, Madhya Pradesh, Telangana, Andhra Pradesh, Karnataka, Jharkhand, Odisha, Bihar, UP, and Uttarakhand. And also there is report of H5N1 outbreaks in painted storks and other wild birds in the Delhi Zoo. And very interestingly, this was again spilled over to the tigers and leopards in Maharashtra in Goregaon wildlife rescue centers. These animals, especially the tigers and leopards, they were shifted to Goregaon in Nagpur, and they were infected, and they are tested positive. And very interestingly, these, and also domestic, these tiger, of course I will come to next. The viruses all were sequenced, and if you see, they grouped into two major genetic groups. One is a Clade 2344B and 2321A. If you see all the poultry isolates, they belongs to this group 2321A. And this virus 2321A is, interestingly, it is not a new introduction in the recent ones. It is since when we compare, these viruses are very closely related to the viruses circulating since 2010 and 11. From 11 in India. That means there is either persistence of this virus, or there is a local migratory birds in South Asia. I am talking South Asia. This virus is persisting since 2010 and 11. 10 was reported in Bangladesh, but 11, since 11, the virus is present in reported in poultry and ducks in India, and these current viruses are related to the 2321 viruses since 2011. That indicates there is either environmental persistence, or there is informal trades between the neighbors in SAARC countries or South Asian countries, or local migratory birds moving this virus from one place to another, and also maybe the movement of animals, this unrestricted movement of birds. Then the Rajasthan virus, that is a new virus, and it is a new introduction into India, though since they are very closely related to the viruses circulating in current times in Eurasia, Africa, and Americas. So these are the risk factors for influenza virus spread. There is wild bird movements. Everybody knows, I think, in this audience, that there is winter season migratory starts from north-south directions, from Arctic region till Indian Ocean, covering at least more than 20 countries, and they stay here for from October, November till February and March. Then they go back. during this time they they come across different countries having avian influenza outbreaks, and so they also act as a risk factor. There are a lot of wetlands. At least 80 wetlands have been reported, Ramsar sites or wetlands, where these migratory birds come and stop over, have feed, and then they mix with the local birds, specifically the ducks. Those are in the rice fields, because they migrate to the rice fields, and where the local birds like ducks, they come and breed. They act as a breeding species between wild birds and the domestic poultry. And then we have a huge poultry population, like 851 million as per this livestock census. And there is backyard poultry where there is low or no biosecurity practices. We have live bird markets and market chains where also there is low or no biosecurity. They also play important role in the mixing of these viruses and spread. Once this market is closed, these viruses, these poultry again go back to the owner place and they stock there. So there is a cycle of movement of these birds without biosecurity measures. Then domestic ducks, they don't show any clinical signs, very few clinical signs, but compared to chickens, but they carry the virus without showing clinical signs, and even they shed the virus up to 7 days without any clinical signs. They also act as a risk factor. Then we have international boundaries which are very porous, and there are reports of informal trades between the borders. So they act as a risk factor. And these are some of the outbreaks reported since 2006, around more than 450 outbreaks. And specifically the outbreaks are, if you see the lower one, the panel, the outbreak starts in October and ends by April, but peaks at January. So during cold weather, the outbreaks are very high. Of course, there are deviations we have reported, and there are two major subtypes India has reported, H5N1 and H5N8, in different time periods. More than 450 outbreaks. They are all resolved. They are not continuing outbreaks in 25 states. That means except few states in south and then some northeastern states. Except that, almost all the states they are infected. They are susceptible. They are infected with the influenza viruses. So we have done some antigenic analysis between the clades, because we say they are genetically different clades. Whether they are antigenically similar, whether any vaccine with one virus can give cover and serologically to other viruses or not. We have done some study with 4 different clades. Antigenically, they are genetically different. So we wanted to see whether they are giving any serological cross reactivity for as a future vaccine preparedness. We see that these clades, though since they are genetically different, they are also antigenically divergent. That indicates that a virus with one clade may not give serological protection if a vaccination is done to the other clades viruses. That indicates. So this indicates. Of course, these are not the recent viruses. We have not done that, is already undergoing. But we see the clades are also, one clade virus does not give cross protection or cross serological reactivity to other clade viruses. That indicates a vaccine with one clade may not give protection, sufficient optimal protection to the viruses with other, if infected with the other clades. These are some of the important informations. If there is a, suppose there is 2321A outbreaks in poultry, and we have a vaccine with 2.2, then if you have vaccinated your flock with 2.2 viruses, and there is emergence of 2321A virus into that flock, then the vaccinated birds may not be protected or sufficiently protected. I cannot say now, because they are serological done, not in the challenge experiment in the field or laboratory settings. So they may not give optimum protection. they may have antibodies, they may reduce some clinical symptoms, but they may not, we don't know, they may not give complete protection to the 2321A viruses if vaccinated with 2.2 viruses, with 2.2 antigen. H5N1, because they are all genetic, since they are RNA viruses, they are.
Vaccines be made for AI.
Yes.
There are so many, there's H5N1 and H7 also, and so many clades should be there.
Yes, luckily we don't have H7 virus, at least we have H5 virus, and within H5 then we have H5N8 and H5N1. Of course, HA only gives protection, not the N1. So H5, in H5, HA is plays an important role. HA antigen is the dominant portion, antigenically dominant. If HA is antigenically close, then it will give H5N1 and H5N8 also. No issue, because N1 is not giving any antigenically coverage. Only HA is dominant, which the antibodies which gives protection, serological protection to the H5 subtypes. But since within this H5, clades are again diverging HA genes, so the antigenic portions also they diverge. Say for example, a theoretical example, vaccination with the 2.2, they have antigenic epitopes of 30. Say example, just theoretical I am saying. Since it is a different clade antigenically, so naturally there will be some difference in the amino acids or proteins. So if they are having 15 amino acids matching, but 15 are not matching, then the antibodies of 15 of 2.2 will not give protection complete to the 2321 viruses if it is vaccinated with 2.2, infected with 2321A viruses. What I want to say, that I mean by that, that if vaccination with the 2.2, 2321 virus will not be protected by 2.2 vaccines.
This particular antibody antigenic diversity, 2.3.4.4b, whatever the recent isolation.
Yes, yeah.
Is whether that was studied in these cases.
That we have not seen, but I will show the international data for that, because there is also international data with 2321A, which is now currently, and also 2344B, which is globally again spreading. That data I will show, but I don't have data from the NIHSAD, but there is a global data is already available that I will be showing here also. I have brought that also, because now the 2344B is the globally dominant strain all over the continents. So
In China, even it has gone to which also, ABCD.
yes, they have different subtypes, but China is different because they have robust system of surveillance, and also they quickly change the antigen. They have used more than 4 or 5 strains now, H5 strains, and they have the reverse genetic systems. As you are asking, I am going a little bit ahead. They have the RG systems where they can change the immediately with HA gene. If it is not matching to the field viruses, the antigens, they immediately they can change the reverse genetics vaccine and they produce vaccines. I have interacted with the, within 1 month they can go into another, jump into another RG vaccine. They have the capability, and also they are doing the surveillance that mechanism. So now it is not one, there are 5 subtypes. Even based on the province they are vaccinating. If this province is dominated by a subclade, they are vaccinating with that also. So they have that, and they are the largest user of the vaccines. 99% of the vaccines they are using. 100% vaccination government funded. All the birds compulsory vaccination. That is they are doing in China. So that is different. Since you asked that, just you can interact, no issue within me. Yes.
How often these clades will evolve?
Uh,
You said like suppose if you vaccinate a crop with 2.2, and if the crop is having a different virus of 2.1, do you think that will make sure that they
there is no point in vaccinating with 2.2. There is a need, since as per my data, NIHSAD data, whatever the NIHSAD data, whatever the states are submitting the samples, based on that I am talking. Majority of the viruses from the 10 states outbreaks we saw, 2321 is the dominant strain, and 2344B it is only in the wild birds. We have not reported. Only earlier in 2024 we have reported in Kerala in ducks. But besides that, in 2025 only 2321 is the dominant strain, Suppose you vaccinate with 2321A, say for example, we don't have vaccination policy as a theoretical 2321A, then if you have 100%, 90, 80% at least coverage of the poultry population, and with good vaccine, matching vaccine with current strains, because it is already 10 years old, the virus spreading. If you have a current recent virus which is having a good antigenic coverage with the viruses circulating in all these states, and you produce a vaccine and it is having, and you vaccinated more than 80% of the poultry population, that is very important, and some good amount of antigen you have given, then it will produce good antibodies. Only thing in vaccine, influenza vaccines, it will reduce, it will stop the clinical signs, but the virus circulation will be there, because it will not stop the infection. It will stop the clinical signs, reduce the shedding, environmental contamination will be reduced, spread will be reduced, but it will give protection. But even vaccinated, you have to go for surveillance. Vaccination is not the end. Vaccination is the starting point of control of the disease. After following vaccination, you have to go for monitoring of the antibodies, whether sufficient antibodies there or not, whether virus is still persisting or not. You have to go for that. Suppose in case in the vaccinated population virus is still detected, you have to call the poultry birds, because since they are the zoonotic viruses, you cannot allow the viruses to replicate or remain in the poultry and have interaction with the human beings, because these are zoonotic. Again, in the presence of vaccination, if there is virus in the vaccinated poultry, the calling policy, you have to call the birds so that the virus does not spread. Only vaccination has an advantage that it will not allow the virus to spread drastically. It will not allow the virus to spread to the environment. The environmental contamination will be reduced so that it will not spread widely like in other. But in vaccinated, if there is, so this is the some of the situations, because you cannot control the disease in isolation. Like India, we can control the disease. Since it is a transboundary disease, we have to consider the neighboring countries also. And if neighboring countries having a virus also, you have to be prepared for that. That the virus can come anytime, because it is through anything it is coming, through the informal trades are going on through the neighboring countries, through migratory birds, or through other routes also. Cultural practices, people take the fighting cocks from one place to another. So these are cultural practices. So these are this is the worst affected area in the South Asia, in this globally, and these are some of the outbreaks reported. More than some 1800 outbreaks reported since, and still this virus is continuing reported in Bangladesh and also Nepal. They have reported. And this is the situation in the avian influenza in the SAARC countries. There are different 2321. If you see, they are the dominant strains in South Asia. In Bangladesh, in India, and very earlier it was in Nepal, also Bhutan. They have reported 2321A. Then there is a different virus 23 earlier also in the Bangladesh, and different time periods, different viruses are emerging, and then they are also going out, and new viruses are coming. So this is the complete, and this is very much important when going for vaccination for control policies. We should know about the viruses circulating in neighboring countries, so that we should be prepared for that. And this is the global scenario. The virus emerged in 1996, the currently circulating virus. Then it remained in Southeast Asia till 2005. From 2005, it spread to globally. By 2006, it has covered almost Eurasia, Europe, Asia, Africa, and the Middle East and the South Asian countries. Currently, the virus which is called the 2344B virus, that emerged in late 2020 in Europe, specifically in the Netherlands, and then it is spreading almost, except the Pacific regions, or the like Australia and New Zealand, almost all the continents, including Antarctica. This virus has spread through the wild birds, and also in other regions to the domestic poultry. and it has spilled over to mammals, 43 mammalian species in Europe, almost all the continents like red fox, mink, fur animals, sea lions, seals, cats, tigers. There are so many 43 mammalian species here jumped, including the dairy cattle and goats and also sheep in United Kingdom. And the current wave, these are the outbreaks reported so from poultry. These are number of outbreaks and wild bird outbreaks across all the continents, except these are some of the HPI outbreaks of different subtypes. They are local subtypes in this region, Pacific region, but almost all the continents. In Antarctica, in early 2024, the virus was detected in wild birds. So this is the first time USDA reported spillover of H5N1 virus to dairy animals, but these virus is completely different from the poultry or other birds. These viruses only found in cattle and some wild birds, these genotypes. These Eurasian viruses mixed with the American some genomes. So these are very interesting. This is the genotypes we are telling B313 and D1. These are the specific genotypes, and they have some mutations which are of human concern. They can jump from PB2. If glutamic acid to lysine, they can jump, I think. So dairy cattle, and also this virus also spilled over to though there is a risk assessment by WHO, they are very low till now to human beings. But those who are involved directly with the dairy animals, they have they are highly they are risk to this disease. And there is report of report in the sheep also. And in India, we have surveyed some few states like Kerala and Odisha, specifically in the infected areas, within 1 kilometer infected area of poultry outbreaks, and we didn't find any influenza virus in dairy animals, including cattle, sheep, goats. We have screened.
So this is reasserted virus or?
They are reascertain virus in the dairy cattle. Whichever the virus was in the Eurasia, Europe, it is spread to North America in 2021. Then 22 it went to South America, and by 24 it has gone to the Antarctica. But these viruses which went to North America, the same virus, they are not the same viruses. They have reasserted with wild bird viruses of the North American wild bird viruses, and these viruses are thought to be died the wild bird by the environmental contamination, these viruses, because in poultry you have the interaction with wild birds, but how these dairy animals are getting the infection from the wild birds or poultry, they are differently placed. So wild birds death was reported in the dairy animal barns or the sheds, and environmental contamination is proposed to be the source of this virus to the dairy animals. And from one animal to another animal, transmission is through the milking. They are milking through the machines, because they don't have any option of getting milk in between. The dairy animals, they are milked through the machine. It goes to a chiller plant, and then the tanker comes and takes the milk. So they detected some thickness in the milk, like colostrum in the initial stage of calving. So when they are doing the serosurveillance, they had antibody positivity. Not that immediately they got the detection. They have a robust system of surveillance. So initially, since they have a serosurveillance system in the cattle, and they have some milk, abnormal milk, they are getting colostrum-like milks. So during serosurveillance also, they got antibodies positivity, and then they tested the milk and swabs of oropharyngeal swabs, then they got the virus. So this is the global distribution of the clades from last 3 to 4 years, 22 to 25. The pink color is mostly the 2344B clades. All the continents, if you see, except the Australia and the New Zealand, all are having 2344B viruses in poultry and their domestic birds and other birds like the mammals. But this is again other clades during the same time period. If you see the India, they have the 2321A and also 2344B viruses, and China have also different 2321, but different again sub clades groups. If you see the clade 2344 virus of 2024 and 25 are suitable for candidate antigen for inactivated whole virus adjuvanted vaccine targeting these strains. Whatever the recent viruses of 24-25 of 2344B, they are antigenically very matching with the previous last 2-3 years. So they act as an antigen for the vaccine. But antigens outside 2344, including 2321 viruses and earlier clades 1 or 221 or 234, so limited antigenic cross reactivity and are unlikely to offer effective protection against currently circulating 2. That means if it is beyond the 2344B, then other antigens like 2321A or clade 1 or 221 or 234, they don't give antigenic cross reactivity with the currently circulating viruses. So coming to the control measures, traditionally, if you see this is the control measures followed, like culling, elimination of the infected flocks, biosecurity measures, you know all this, how to keep your flocks, your healthy flocks from the infected flocks or the infected viruses not coming to the by physical barriers, movement restrictions, because you know if the infected viruses you move the poultry, then it will again infect the other viruses. Diagnostic and surveillance. Surveillance is very important to say whether the virus is present or absent. You have to go for surveillance. Then education and training to the poultry workers. If they don't know how the virus spreads from one farm to other, then that will create problem. So beside all these 28 epizootics, only 5 epizootics vaccination has been practiced in Mexico and in Pakistan and North Korea and again in Mexico, and the current outbreaks, the vaccination has been followed. But vaccination should not be considered as a in isolation. Vaccination, if you go for only vaccination and then you forget, then it is not going to solve. Control measures, this is act as a additional control measures, or it should not be used in isolation. It is another layer of protection you can consider. Besides biosecurity, vaccination is another layer of protection, additional layer of protection, and this should not be considered the last point. This should be fast. If you once go for vaccination, you have to survey whether vaccine is working or not. And sometimes in the presence of vaccine also virus diverges if it is not optimally vaccinated or there is no optimal antibody level in the flock. And international organizations also, they have the policies and guidelines for vaccination, because these are in 2023 general session of OA. They have this policy documents they have brought out. Avian influenza vaccination, why it should not be a barrier for safe trade, international trade. It should not be a barrier. As per the OA guidelines, vaccination is not a barrier for trade, but you have to follow the OA guidelines. If there is vaccination, you should have a robust surveillance system to say that your exporting countries, the importing countries, that you in the vaccinated flocks there is no virus circulation, and the antibodies present are due to vaccination, not due to infection. That once you prove, then it won't affect any international trade barriers or restrictions if you have good surveillance data to supply to the importing countries. So this is some of the merits of the vaccination. It increases resistance to AI virus infection. If you are vaccinated, then if it needs 10 to the power 6 dose, then it may need 10 to the power 9 or 8 doses. It will resistance. It will have resistance. Very quickly it will not be infected. It will have resistance to the infection. reduce AI virus replication in respiratory and GI tracts. Naturally, when there is antibody, naturally the virus cannot replicate very fast. Prevent disease and death in poultry. It will does not show clinical signs and it will not kill the birds, but the virus spread will be there. It will reduce the leading to a reduced environmental contamination, so that once contamination is reduced, the spread will be also reduced. Then reduce spread between farms, reduce transmission to birds, then improve animal welfare, because once the virus infection is there, you have to call it. And if the virus spread is low, then naturally you are the animal welfare, you are not calling the birds or they are dumping. But as I told earlier also, it adds an additional layer of protection on top of the other biosecurity, but it does not replace biosecurity. You have to follow biosecurity measures and hygiene practices in the poultry farms. Some of the experience of vaccinating countries. China substantially decreased the number of H5N1 outbreaks. Of course, China went for vaccination since 2004. Of course, by 2004, the virus is already endemic in China. So, following endemic, they went for vaccination. Of course, 100% vaccination in China. They have emergence of divergent strains, because again, when going for 100% vaccination, maybe they have the divergent strains emerged in China, and also Egypt they have reported. The vaccine coverage is an issue really. If you see in Egypt, 25 to 30%, that is the best vaccinated government farms, while as little as 1% coverage in villages against requirement of 80%. If you don't have a flock antibody protection of 80%, naturally the virus will spread. But this is the funding requirements, use of technical manpower. This is required for vaccinating, and then how much cold chain and logistical support. These are for vaccination. These are required for a country for covering the poultry populations. So less than up to 1% they have vaccinated. So naturally, where requirement of 80% and you have 1%, so naturally, in the presence of vaccinated birds also, naturally the virus will be diverged, because there is no complete or optimal coverage. Technical incompetencies, biosecurity failure leading to mechanical transmission. These are some of the examples in some of the countries. Though they are vaccinated with the same syringe, maybe though they are harboring or the virus with the same syringe and needle, they vaccinated the healthy birds also. So instead of protection, and you are infecting the birds while the vaccinating. So these are the technical incompetencies. Some of the countries they have published it, reported that the vaccinator, they don't have the technical competency. With the same needle and syringe, they have vaccinated with one farm and they are going to the another farm. You never know whether the virus is already harboring in that poultry farm or not. So through that also they have transmitted the disease. So these are technical competency required. Then economic impact. This is actually a publication from the China actually. If you see, they have some mock actually testings and simulation exercise. Based on that, they are telling if you go for culling vaccination, the culling will be 1.36% of the cost of the routine vaccination. They want to say that if you go for culling of the birds, it will reduce the cost than the vaccination. There is only a simulated disease model, not a practical one. Then it is again labor intensive. Vaccinating individual birds by pricking is labor intensive. Then these are some of the vaccination history countries. At least 14 countries vaccinated towards HPI 2002 to 2010, like China, Pakistan S7 vaccines, and then Mexico, Cote d'Ivoire, Egypt. These are the countries they have gone for vaccination. Different way of vaccination, preventive vaccination, and then emergency vaccination, and endemic or routine vaccination. And these are the percentage vaccines used. Like if see, more than 99% vaccines were used by the China, and Hong Kong also 100% vaccination. And these countries only went for preventive vaccination, emergency vaccination. And during 2002 to 20, more than 450 billion doses of vaccine used, approximately 25 billion per year, mostly inactivated vaccines. either it is reverse genetic, mostly it is reverse genetic vaccines, because whole virus or field virus vaccines, inactivated vaccine is not, it is not advised, not allowed, I cannot say, it is not advised, because Indonesia is going for direct field virus strains and inactivating, inoculating. No other country is doing field virus, inactivated vaccine. Others inactivated vaccine, it is reverse genetics virus. From HPI virus to LPI, they have converted and produced a LPI virus, and then gone for, because of spillover and also the containment facility, requirement of containment facility for vaccine manufacture, because you are handling a high path virus. So through reverse genetics, the vaccine has been reduced to LPI low pathogenic, removing the basic amino acid from the HA gene, so that it becomes low pathogenic. Then the virus is inactivated and inoculated. So this is the difference. Both are inactivated vaccines. One is converting the high path field virus to a low path virus by reverse genetics, and another is a direct field virus inactivation. International organizations don't subscribe to use of a high path field virus, because you are amplifying the virus in an environment where the people are working, and there is chance of spillover, environmental contamination, which is again a zoonotic importance. These are some of the vaccine technologies. We have the inactivated whole virus vaccine produce only humoral immune response, whereas the the recombinant vector vaccine produce cell-mediated mucosal immunity and also humoral immunity. Whereas the inactivated whole virus, whether it is heterologous, homologous, or combination vaccines, AIND or heterologous, like for H5N1 virus, you are going for H5N2, because HA is both common for both. For homologous vaccines, H5N1 vaccines, for H5N1 strains, like in Pakistan, they have used the inactivated vaccine. Indonesia also, they are using directly field virus, inactivating and inoculating the poultry. And then another is your reverse genetics inactivated vaccine, matching the field strains. Specifically, China is using this one. They are they have the surveillance system. They know what is the virus type circulating and what is the vaccine strain now. They are matched with the sequence data. If the vaccine strain, the field viruses are diverse from the vaccine strain genetically, they go for selection of a new strain. And then that new strain, the HA is inserted in the reverse genetic system with all 8 genes having, only HA is replaced with the current viruses, and then they go for producing a LPI virus, which is again inactivated and used as a vaccine. I think that is clear what I am telling, reverse genetics and field virus vaccine. Then combination vaccine also, AI and Newcastle disease also people are going. Currently, you must have heard that France has gone for vaccination by government funded vaccination. Around 85% of the funding is by the government and 15% by the industry. And now second year they have again reduced the government funding 75 to 25%. So that initially they want to see whether the vaccination is working in the field condition. Only the broiler ducks they have gone for vaccination. This is the third year of vaccination. They are doing vaccination with the combination AI and ND, but they are monitoring only AI. These are some of the recombinant vector vaccines, like fowlpox virus with HA gene insert, then herpes virus turkey, Newcastle disease. This is DV. I'm forgetting that. Just duck enteritis virus. Yes, duck enteritis. Thank you. So these are the some of the vector vaccines used with HA gene inserts. So they have a good protection of cell-mediated immunity, mucosal immunity, and as well as humoral immunity. Then there is a need for DIVA strategy, as I told, for trade purposes. You have to say these antibodies are due to vaccination or due to the infections. the strategy may be sentinel poultry or serological testing through different methods available already, because your protection is HAGIN. So if you have a outbreak of H5N1, then you can go for a Diva vaccine like H5N2, so that you can monitor the antibodies are due to N1 or N2. If it is N1, then it is infected. If it is N2, then it is vaccinated. So same way also different non-structural proteins or the nucleoproteins can act as a control to know whether it is antibodies are due to vaccination or due to infections. Then trade implications already I have mentioned. If you have a good surveillance system in place, and you can say that the antibodies are due to the vaccination, not due to infections, and as per this provision of OA code, if it is there is place, then you can export. International trade poultry can do. Then responsibility of the countries going for vaccination should have high quality, reliable, registered vaccine matching with the circulating strains. Matching is very important. If it does not match, you are vaccinating, but still there is virus circulating and spreading. Surveillance capacity, you should have a surveillance and monitoring capacity also. Following vaccination, you should go for monitoring whether optimum antibody is produced due to vaccination or not. Any virus circulation following vaccination, that also need to be monitored. Then poultry producers' commitment, they should adhere to the disease control measures specifically very clearly. Even in the vaccinated poultry, if there is virus circulation, the poultry population flock needs to be called and disposed and sanitized. Data collection from producers and veterinarians, these are very important to know whether the vaccine is really working or not. Is there any need for changing the vaccine strains? This is important. Then capacity to ensure traceability of the entire process. Again, that is important, from vaccine production to on-farm administration, post-vaccination monitoring, including traceability, animal and their products.
The vaccines that are being produced, inactivated, is it mandatory to have SPF-X or embryonic eggs?
I could not follow. Vaccines already produced.
The vaccines intended for AI.
Okay.
The production is done on
Achha, through
SPF-X
SPFX or okay, I understand. That means growing the virus, naturally it should be SPFX or the specific antibody negative. It should be at least the antibodies in eggs, you can detect the antibodies in the yolk. The antibodies should be at least, should not be same antibodies of, suppose you are producing H5N1 vaccines, the antibodies should not be H5N1, because already antibodies are there in the yolk. If you produce, it will not grow the high titer. So it's ideally it should be SPFX. If not, then specific antibody negative X, embryonated X if available, then it will be ideal, because you need a good titer. If the antibodies are there, naturally the titer will be reduced. Your main aim is vaccine producer, a dose. When he is producing a low antibody titer, he can double high antibody, he can double the vaccine dose, 1 million to 2 million if he dilutes twofold. If the titer is very less, then he cannot dilute. So it is again the dose matters, the titer. At the end, it is the titer of the virus in the allantoic fluid, infected allantoic fluid, that matters. If it is high titer, then naturally it will have a good number of doses. You can dilute it. So that is important. Or else, if you don't have the SPFX, then you can go for specific antibody negative chickens, eggs from that.
So one more, is it mandatory to have these vaccines produced in a separate facility, or can it be produced along with other vaccines?
That is a difficult question actually. I don't know, but like if the combination vaccines, whether they are producing the same facility or not, I don't know. It is up to the how GMP and the other things being maintained, because the contamination of your seed stocks and other things, you have to see that. you should not contaminate your seed stock or other. That part I don't have much idea. Actually, the production part I don't have. Uh, Even field experience, I don't have much idea on the field exactly. If you ask me, very limited experience of the outbreaks, very limited, or expert in the laboratory conditions, very limited experience. The current control strategy, yes, I have done that. This is the strategy followed by the Government of India, detect and culling policy for HPI in domestic, cleaning and then disinfection of infected premises, then burial or incineration, restriction of movement of poultry. Naturally, that is very important. From the infected zones, 1 kilometer is the poultry should not move, or any material should not move from infected area to the any healthy areas or the surveillance areas, including the poultry and the poultry products, movement restriction. Then compensation to the farmers on the forced culling, because we are within 1 kilometer. If one farm is detected positive, then within 1 kilometer, the police says all the birds needs to be called, uh, then buried and then disinfected. So forced culling of the birds, compensation is to pay to the farmers by the state and the central government. Some maybe ratio I don't know, but they are paid. Then POSP surveillance within 1 kilometer area, all the poultry products and materials are there buried, burnt, and then sanitized. Then sanitary certificate is issued by the state government in consultation with the Department of Animal Husbandry. Then there is a surveillance, post-operative surveillance plan from 1 kilometer to 10 kilometer area for 4 quarters, 15 days interval. Collect the samples from all the villages within 1 to 10 kilometer, tested it 4 times. If it is negative, then it is opened for restocking the birds. No vaccination till now in Government of India for HPI. We have a surveillance system for controlling this disease, both active and passive surveillance as per this Government of India action plans, passive and active surveillance of wild birds, domestic poultry. They are a continuous activity of state and union territories, and already sampling strategy is already given in the action plan. Till up to the district and district level, districts have been identified, and how many number of blocks and poultry to be sampled has already been mentioned in the action plan. This is the how the flow of information from through the surveillance network. The samples are collected from the unusual mortality or sample collection sites by the sample district investigation officer or chief district of veterinary officer, CBO, or district epidemiological officer through his field functionaries, and then information goes to the state animal husbandry directors. The samples goes to either the RDDLs. These are the RDDLs. We have 6 RDDLs, and this is the central laboratory, NIHSAD. If there is heavy mortality, you consider there is high mortality. To get the results very quickly, directly the samples are sent to NIHSAD, and then these results are communicated to the Government of India, and also a copy goes to the state director and chief secretary. If there is a routine surveillance, the samples may go to the RDDLs within their jurisdictions, tested. If they are positive, then the samples flows, positive sample goes to the NIHSAD with the results, so that confirmation and repository maintenance is done. So these are some of the RDDLs: Jalandhar, Pune, Bangalore, Calcutta, Guwahati, Bareilly, and ICAR-NIHSAD, the central laboratory. So this is how the sample and information flows. So this is the district investigation officer or district animal husbandry officer, or CBO. He is the responsible officer for collection of samples through his field functionaries, the veterinarians, from as per the guidelines. Then these are the surveillance: clinical, virological, and serological surveillance. Like we collect the samples for virological testing, serological testing from the districts, and has been identified. These are the samples collected from poultry and wild birds, swabs, environmental samples, water from water bodies, dead birds or face fecal droppings. These are the samples collected. Then we have also gone for limited surveillance following the detection of H5N1 dairy animals in these two states, Kerala and Odisha. And these are number of epicenters, cattle, some and buffalo, goats, milk and nasal swabs. They all found to be negative for influenza A virus. Through this active surveillance, we have detected some H5N1 viruses, which is very much persistent in India. As per the industry, it is more than 6,000 crores loss to the poultry industry, as per the poultry industry statements. So, this virus is circulating in different poultry farms, and the virus has been isolated from poultry, dogs, and from the environmental, from the live bird markets, and they belongs to one group of viruses. And NIHSAD has developed a influenza vaccine against the for H5N1 for chickens based on the based on this sequence data available from 2003 2000 till date. And we are still going on vaccine matching, whether the vaccine strain developed is matching with the current viruses. It is an inactivated vaccine, and it is already commercialized. Then, as a preparedness, NIHSAD also working on vaccines, reverse genetics vaccines, RG H5N2 vaccines. This is as a research. We have this is like a high pathogenic HA, multiple basic amino acids converted into low pathogenic. This is the basic amino acids, multiple R, lysine and arginine basic amino acids. We have converted only one basic amino acids, and this is from India again, and H5N2. So there is antibody production till 200 days with 2 doses of vaccines. There is virus. There is virus shedding, though there is virus shedding different time periods, but there is lot of antibodies, protective antibodies remaining for 200 days of the study period. This is some of the recommendations for poultry farmers, like implementing strict biosecurity measures, report unusual mortality, separate wild birds from domestic flocks, for which keep the pathogens. Why this biosecurity? Keep pathogens away from poultry and poultry from pathogens. How to keep this? This is the virus. How to keep this by physical barriers? Goal is to prevent the introduction of spread of invisible pathogens, avian influenza. Total structural barriers and procedures to keep the pathogens out of naive flocks from leaving the infected flocks. But why modern poultry production do we still have outbreaks? Biosecurity reduces but does not eliminate risks. So this is the message I want to just show. Biosecurity plays in global strategies. Now there is another strategy developed by OA, FAO and OA, global strategy for prevention and control of high pathogenic avian influenza, which focuses on sustainable poultry production system, public-private partnership, coordinated One Health approach. One Health then encourages investment in biosecurity, early warning systems, that is surveillance systems in different environmental surveillance, capacity building, transformation of poultry sectors. Coming to the conclusion, it is very difficult based on this current scientific evidence. It is very difficult to eradicate the virus, HPI virus specifically, in near future. So strengthening surveillance in poultry, wild birds and other hosts is important to assess their impact, animal health, and act as an early warning. If you don't have a surveillance system, the virus will be circulating. So this is surveillance, like environmental surveillance, which gives an early warning whether virus is persisting or not. Enhance biosecurity to minimize the contact between poultry, wild birds and their fomites. The vaccine need antigenically relevant strains to match the field virus for consistent protection. Poorly matched vaccine will not provide the predicted protections. Like if you have a vaccine with 2321, it will not give prediction to the 2344 virus outbreaks in poultry. So some of the links, useful links, these are some of the action plans, OA avian influenza guidelines. This is avian influenza matching programs of international OA and WHO, OA and FAO. then CDC, HPI dairy cattle, then we have confirmed case of livestock. So these are some of the useful links I have given. So thank you. Anything?
Thank you so much, Dr. Tosh. In fact, that was very, very simple, but nevertheless, the exhaustive overview on avian influenza, with the special reference to the varied distribution of different types of cats and the subcats at the global level. Then the spillover of this infection from poultry to mammals, which is very much immense concern. And then with reference to the vaccine technology, including the DIVA, and then it was very interesting to learn that in 14 countries the vaccination has already been introduced. Okay, so before I just request my colleagues to take part in the question session, I just have one major clarification to be sought from you. See, in India, very recently, the NIHSAD has initiated to develop both LPI as well as the HPI based inactivated vaccines.
Only LPI, not HPI. HPI were just research. In the research area.
But one of the major reasons why it was not initiated, but it was already there in other 14 countries, is because of very high possibility of interference with respect to the surveillance mechanism, which is already in the place, isn't it? We also spoke on this DIVA vaccines. So DIVA is basically meant to differentiate the antibodies because of natural infection or whether the antibodies are because of the vaccination. Whether these vaccines which are likely to be developed or which are already in the pipeline at NIHSAD, are they working based on DIVA, or do you have any plans to develop DIVA based vaccines so that it should not interfere with our ongoing surveillance mechanism in India?
What I presented, it is already developed, but it cannot be commercialized. That is actually the RG system. It is DIVA enabled vaccine. It is yes. It is DIVA enabled. Only thing, the vectors or the reverse genetic system which was procured from through a maybe research or something. Some of my colleagues, actually I was not the part of that technology. It is some other scientists, they have developed. They got the DIVA this reverse genetic system, but this is for research use only. You cannot commercialize that with using that technology. So,
in these 14 countries where the vaccination is already introduced, none of them are DIVA based.
No, they have the DIVA based. Like in China, they are very much in DIVA based. There is 100% DIVA based, though there are some of the vector vaccines are available. Bangladesh also has gone for DIVA based vaccine, but they don't know whether the vaccine they are using, they are importing from other countries, whether what is there in the vaccine and what is in the field conditions, they are not aware of it exactly, because somebody told that you take this vaccine, it will work. They don't know.
Actually, it was the case earlier, but of late, the case is the government insists every company to have a trial with one of the universities or the livestock institute, and based on the results only they give go ahead for the vaccine or any product.
So they are doing in a experimental setting in a laboratory condition, then doing the challenge study in the experience settings, and then they are releasing the vaccine to the field. Okay.
So this is two children. Those are students only. So I have one question. Like RG system is already there now, right?
RG systems?
Reverse genetic system is already there in our,
yeah.
right? So suppose an outbreak occurs. How quickly we can investigate and take out those genomes and insert back into our backbone and all that?
Like in already I told in China, they told that within a month they can replace.
We have something
No, reverse genetic system we have, but that we cannot use it for commercial, for the purpose of IP. It is IP protected.
For the purpose of restriction of the spread.
Yes, this is IP protected. Yes, this is IP protected. If any company is having a reverse genetic system and they collaborate with a research institute, then because they buy the reverse genetic system with the company, the research institute know the which HA gene is matching to the population. Then it can be incorporated and do that vaccine develop can be done. But as a
No, we
we don't have a reverse genetic system for commercialization because it is IP protected, intellectual property protected, which we cannot go for commercialization. Though we have a virus developed from this system, we cannot go for commercialization of the technology.
So another question is on broadly utilizing it. Is there any work which
Neutralizing.
broadly
Yes, people are working on same way universal vaccines. People are working, but like universal. This is simple. Universal vaccine means which having broad antigenic coverage, broad antigenicity across the influenza viruses. But the protection is offered by HA gene, which is highly variable. Based on that, you have the clades and antigenic differences. And the which is very giving the broad protection is the matrix gene, which is across all the influenza subtypes they are conserved, but that will not give protection because HA gene is responsive for humoral immune response antibody. So people are still working on using matrix and other genes combinations. They are trying RNA gene also, and also HA conserved region. In HA1, HA2 is conserved, but main problem is issue is the H1 is antigenically dominant and which is highly variable. So the issue lies which one to take as a universal vaccine which will give antibody against all the influenza subtypes. Even if it is not all the influenza, at least 2-3 subtypes, because influenza subtypes there are 16 subtypes. We don't have problem with 16 subtypes. We have problem only with the 2-3 subtypes: H5N1, H5N8, and H9. H9, H7 we don't have now in India. Say for 2 subtypes or 2 subtypes. If we can do that, that will be a great achievement. We don't. People are working, but there is no that much success, and there is no. I don't know. Field conditions. Yes.
Maybe Dr. Yashwant, in fact, is planning for something like this. May I ask you to request you to contribute to it?
Thank you, sir. Actually, just recently I got an award from ICAR regarding the universal vaccine. So I'm planning to use mRNA-based vaccine. Yes, because I know that whatever we are doing, as such that is a reverse genetic-based vaccine, but still it's kind of vaccine where we don't have to have our own exact viral particles in it. So I'm planning in that aspect, try to cover all the.
What aspect you are looking at?
mRNA against HAGs.
Influenza.
Yes.
Oh, excuse the question I was kept because
That's why I make it to Dr.
okay, so he is.
Yeah, that is also okay. And we will work together. And so recently we are planning, and I don't know how far we can able to do it, but at least for some extent we can able to find.
Please focus the initiative. That itself is a big.
Yes. So we got it, and it's in the initial stage, and we are planning to. We are hoping that this can protect against 2, 3, 4 weeks. But still, it's not there in India. But we're hoping in the future we can able to have a universal vaccine for all the 3 combined, like people, animals, and poultry. So yeah, so in short, it's still in the initial stage.
He will produce the vaccine. We will do the laboratory experiment.
Definitely, sir. I mean, because NIHSAD is the only place where we can able to access the laboratory and for the vaccine, and their contribution is much more than what we are doing. So in vitro, what we can do it in any of the labs, but the outcome is coming from the NIHSAD. So I'm happy to see today, like we just met. So we've been interacting over the phone and email. And so I do have one question on that. So the LPA which you have produced, we are hoping to get that vaccine technology for register for animal health. And so is the same H5N1 is present I mean, the FDA, what other countries are using. So is it the same H5N1 or are there different clades used?
This is actually there are three lineages globally in Eurasian region. G1 lineage, but within G1 there are sub lineages are there. So this virus is actually from the India. So this is we expect that it will have a broad antigenic coverage. At least it will have a coverage to the Indian strains. Though there are G1 lineage vaccines available already produced by international manufacturers, but we don't have direct trials in animals, so that we import, since we have already a vaccine, Indian vaccine, which is developed based on genetic analysis, based on antigenic cartography, and as I told that if I come to that, this vaccine is developed based on phylogenetic analysis. Whatever the virus strains available in India since 2003, we have compared it, HAGene, and from that we have taken 7 viruses identified from different groups, then raised anti-sera against all the 7 viruses and did a cross serological reactivity, then mapped in a 2-dimensional structure cartography from each other how much antigenical difference, then which is in the center, that strain we took. We selected some 2 strains, again we go for growth kinetics, because you need again the vaccine, you need volume in the, so again from the 2 strains we see what is the best growth kinetics, producing highest quantity of virus within 72 hours or 60 hours, titer highest titer. So based on all this antigenic analysis, genetic antigenic cartography and growth kinetics, and which gives antigenic coverage to circulating strains in India. That is we tried the best way possible scientifically, so that it gives a broad antigenic coverage. So that is an Indian strain used. So and G1 vaccines already available global manufacturers, but that G1, though antigenically may be how much we don't know, because it is a G1, but because within the G1 if you see there are different groups again, whether they are antigenically matching or not we don't know. Again in outside also there may be a highly antigenic genetic divergence. So if somebody wants to use imported H9 vaccine, then we need to study in the animal laboratory, animal conditions, animal system. We vaccinate it, challenge with our field strains. If the field strain challenge is substantially protection, then we can go, or else that is the way how we can evaluate a field, the imported vaccines for our poultry.
Another question, you mentioned about environmental sampling. Okay. So do you think the clade formation can happen in the environment, because the virus can stay for longer time in the environment? And also, like, are there any action plans already stated by government of India? Because some researchers see that sewage water is having they can detect this, and also there are some points also having that. Of course, there are no wild bird intervention. So do you think it's circulating among us, and any action?
No, environmental surveillance, the virus can survive for long if it is a cold climate. If it attains 40-degree temperature, the virus cannot survive. In India, if it is in hot summer, the virus cannot survive. In dry heat summer, in Indian condition, cold climates like in Arctic region, maybe years together virus can survive under the ice. And environmental surveillance already in the action plan, it is there. We are also under different projects also we are doing environmental surveillance, including the live bird markets. That is the best way where there is human animal interface. The poultry value chain, like in the slaughterhouse, poultry processing units, we collect the environmental samples, wastewater, like after the defeathering, they dip in a bucket of water we collect that water and then we test by precipitation methods concentrating the virus. We have at least 25% of the samples we got positivity. Then the knife swabs where they cut, and the cutting board, you have a wooden board they cut in that, and also there is a table, processing table. Then they throw the waste into the offals. So we collect these samples, and also drainage water, we collect the samples and we do this environmental surveillance. Also air sampling we also do, but so far there is no success much. We do the air sampling in the slaughterhouse where there is poultry slaughtering. The principle is you generate a lot of aerosols while processing the poultry, blood and other aerosols. The air sampler just suck the air in that area with different settings are there. In a buffer it will mix for 10 minutes, 15 minutes it will mix. Then you go and with it precipitate and concentrate the virus and test it. Though in air sampling we so far we are not successful in getting the virus, but from the environmental samples, from the wetlands also, from the fecal droppings, we have isolated the virus, detected the virus, influenza virus, different subtypes. Some HPI, some LPI, H9 and H5 also, and sometimes environmental surveillance both, because it is not a living system, it is an environment. So both birds may be processed, H5N1 infected and H9N2 infected. So in the swabs, both the virus you will get. We detected both H5 and H9 from the same sample, from the environment, not from the poultry.
This is with respect to cost effectiveness at the ground level. It was really surprising to learn that culling the birds is more cost effective than undertaking vaccination.
That is a simulation exercise what I showed, because I thought at least to the audience
must have been done based on
some data. Yes, it is a publication. I thought it was just I will show to the audience. People should yes.
So,
but still they are going for vaccination.
Exactly.
They are going for vaccination.
And here the vaccination is only to the parental, isn't it?
Yes, yes.
Any thoughts to make this vaccination strategy more cost effective? Administration of vaccine through the pellets, just like what we have in case of Newcastle disease vaccine, they have the pellet-based vaccines or administered through the water. Any thoughts on that?
No, it is already, I think there are vaccines like the not reverse genetics vaccines, the recombinant vaccines. They are advising for the day old in the chicks also. Yes, and they are advising that it will not even affect the maternal antibodies, because if your breeders are vaccinated, naturally you will get the antibody in the day old chicks, and in the day old chicks at least antibody persists. Field condition you may be knowing, maybe 15 days or 21 days may remain antibodies. If the parents are vaccinated, the breeders are vaccinated, the virus in the chicks, they are already for the 10 to 15, 2 to 3 weeks they remain as antibody. If you, during that time you vaccinate inactivated vaccine, naturally it will be neutralized. So they advise that if you recombinant vaccines given, since it will produce mucosal immune response and also CMI, so it will not neutralize the vaccine. That is some of the companies they are advising. It's already there.
I have a question with LPA vaccine. Reproductive value is only 120. Isn't it
128 HI titer. Yes.
Isn't it
As per OI guidelines, if you see 32 is the protective antibody. If you what I showed, that is for a
LPA.
this is H5N1 LPI vaccine, but that is for against H5N1. This virus is converted into a LPI strain, but this will give protection to the HPI, yes, and the titer is 128. That remains. 32 is protective as per OI guidelines. Yes, what you are talking, if you have a higher antibody, it will have a the protection days will be more.
Any timeline, sir, how long the knowledge protection?
But actually, this is very difficult in the field situation and in the laboratory setting. We do experiments in the SPF chickens, specific pathogen-free chickens, but not the commercial layers or broilers. So the laboratory results and the field results will vary, but it will give some idea. Yes, what you are talking, if the antibody response initially is high, it will have a more longer persistence of the antibodies, but that is you are talking about the for the breeders and for the layers. But in Indonesia, if you see the virus antibody titer, I don't know exactly how much, but they have gone for different types of virus vaccination schedules, like 3 vaccination schedules before production stage, 2 vaccinations, then following 1 or 2, some 2 to 3 vaccination before 19 weeks, 18 weeks of before production, then following production also 2 to 3 vaccinations.
Interval between two vaccines.
It is around initial they have studied by 8 days, then 18 days, sorry, 20, that is 28 day, 2 weeks interval, like that they have given different time intervals. They have seen the number of vaccinations gives better protection, but again you have to see the cost again. If you go for vaccination of 7 doses or 5 doses, till 21 days or 15 days you have antibodies. If you vaccinate and by 35 days the poultry is out, and then vaccinating in 20 days or 21 days, again vaccinating immediately antibody production will not be, it will take 10 to 15 days again. So how much vaccination you want to do in layers or breeders, that is depends again.
So, I'm Dr. Prakash Reddy from Microbiologicals. Thanks for the update, what we have done. So, to update to the audience, we have collaborated to get the technology transfer to produce the H5N1 vaccines, low pathogenic avian influenza vaccine. We have a licensed vaccine which the technology has been transferred to ICAR-NIHSAD. Bhopal. Okay. So, that is in the field now. It is almost 10 months now. We are monitoring all the flocks with whether the vaccination has been done, and it is giving a better control over the LPI vaccines. Okay. But definitely it's a moving target. We need to have a proper surveillance to know how the virus is changing. I feel that there is more of reassortments also which is happening in the field. I think you can throw some light on that. So the reassortment between the H5N1 and the H5N2. There is a lot of strains between. There is some NIV articles also showing some sort of results. What is the, in the sense, how we need to focus on that reassortment? Whether we should focus only on the HA gene or other genes also may have some impact on the prevalence of the organism.
The reassortments you are telling, the H9 virus is contributing to the H5N1 virus, not the H9N2 virus, H9N2 virus by H5N1 viruses. It is the H9 viruses contributing to the evolution of H5 viruses. When since it is there is no concern for H9N2 vaccine, because the H9N2 vaccine is not getting reasserted. The H5N1 vaccine which is circulating, it is getting reasserted by the H9N2 virus, not the vaccine, by the virus, by the internal genes only. But the vaccine is protection is offered by the HA gene. HA gene is not getting reasserted. The internal genes of the virus is getting reasserted. And specific on for H5N1 virus, for H9 it is not affecting. No, it is not affecting the H9 virus. The H9 is contributing to the H5 virus evolution. Yeah. H9 also may be, yes, yes. Yes, both the viruses important, but we don't have a vaccine for H5 official.
Just a last question from my side.
Recently, we are hearing, you also mentioned about cattle being prevalent in the pig, and we are hearing a lot of incidents from the US. So, till now we have heard that the human cases, the transmission could be very less. But is it likely to be transmitted even through milk, or what is the future, like from a point of zoonotic and coming to human? That's the question, because this we are hearing this public scare a lot.
Yes.
Already.
There are a lot of, at least what I from the literature, there are 70 human infections in USA. They have reported, and those who are really the majority are working in the sheds in the domestic animals. They are getting close contact. They are getting the virus and getting infected. But there is advisory that still the virus is it is the risk is low, at the low level. By the WHO, they have done a risk analysis. Based on that, it says that the risk is low for the general public, like we people. We are not going to any farmer, but those who are working with the animals, the risk is a bit high, and they should take precautions like your PPE, protective gears. Those who are working in the barns or farms with the animals, and so they are high risk people. They should have take the personal protective equipments while working in the farms. So that is the only. But so far there is no human to human transmission is very limited. Not that means I want to say there is no report of human to human transmission. We don't know whether there is there or not. There is the pandemic when starts when there is one human to another human spreading. The R value is more than 1, but there is no human to human transmission. Very limited human to human transmission reported earlier, very earlier before 2007 in Vietnam and other Southeast Asian countries, in a family cluster only. Those who are working in the poultry farms or poultry slaughterhouses, and they are in the same house with their children or with their parents. So that cluster, family cluster infection has been reported earlier, but recently there is no human to human transmission. So the risk is low to the public, general public.
Part of my question was already hijacked by. So, in continuation of the same thing, so for a virus to jump from one host to another host, perhaps it was not that not expected that it would jump to the mammals. Although some kind of hypothesis was there, and we are seeing the reaction different mammalian host species which have picked up the infection from this poultry. To what extent the genetic profile is going to change? See, to jump from one host to entirely different kind of host, it will have to undergo some kind of genetic changes. And then whether there was any changes to the extent that it has changed itself, and some light on that, Tosh, or whether it remains the same.
Some cases there is change in the genetic markers. In some cases, like in cat in India, there is no change in the genetic makeup, because if you see the scenario how the cats get infected, you must have observed. I have observed that because I go for environmental surveillance to the poultry slaughterhouses. If you go to a chicken shop, you are buying 1 kilo of chicken, then he will ask, give me 20 rupees these offals. They keep the offals in one side. They take 20 rupees offals or 50 rupees offals. What for they are taking these offals? They are feeding mostly to the domestic pet animals. Either the pet means 2 pet animals, we have a dog or a cat. So we have not directly linked, but when we compare genetically, they are very closely linked to the poultry viruses and with the cats. From the Madhya Pradesh, when we compare that the cat virus and the chicken viruses we got, they are very similar, almost And also the another scenario I am telling, like the canines in the zoo, we provide them the meat. Per day, at least 7 to 8 kilo or 10 kilo meat they consume. Maybe for 1 week they give fasting. I think that is the way they feed the canines. I hope one day they give fasting, some day they decide and they give the meat. And meat is mostly the beef. Beef means it is buffalo, not the cattle. Beef is buffalo. But the again again the virus isolated from the tiger and leopard, they are related to the chicken viruses circulating. So I am not directly saying, but this there is a link with epidemiological link, I can say by the scientific words. The viruses circulating detected in canines or large canines in zoo animals and the chicken, they are similar. So indicates the epidemiological link. So how you link it is up to you, study it, the link, but they are related. And we have so this is again the wild birds when they are dying, the sea mammals and the sea seals, and they eat it. The carnivores, the red fox, and they eat the environmental, they pick up from the environment, you can say directly they consume all this. And the crow again, you see, crow, they are scavengers, they are all peri-domestic birds. They maximum fly 10 to 15 kilometers only from their roosting site, they don't go beyond that. They stay in the human population only for food. They pick up the viruses from the crows and the chickens, they are very similar. So that indicates the virus, though there is no, and we can extrapolate it to that, they are the indicators of silent spread of H5N1 virus. When crows dying, there is no chicken death, we say there is no sir, there is no mortality in chickens, good, but the crows are dying. So we know it, they are the silent, they are the indicators of virus spread. They are picking up from the environment because they are the scavengers. They stay in the environment, they stay in the human population for food. So they consume what they consume, we also know it. So they pick up from the environment and they die. Jharkhand 2011, October and November, thousands of crows died in Jharkhand. And they are related to the chicken isolates. So they are picking, and some cases there is a change in the receptor specificity. Some has been reported, like dairy animals, some percentage of dairy animals there is change in the receptor, or to jump from one host to another host, specificity, signature residues specificity has been seen. And like in humans, those viruses, chicken viruses going to humans, there is clear some differences of human adaptable residues identified. So there is
protease enzymes of human origin also, unless you have to cleave it from its precursor to the
no, they are in the receptor sites actually, human receptors, like the bird flu viruses, like there are different receptors. We have also the bird receptors, but H5N1, but they are in the lower respiratory tract, not in the upper respiratory tract. So the virus, how the virus is going to the humans, if you continuously expose to the virus in the slaughterhouse or in the poultry farming system, the virus will go deep inside the lower respiratory tract, where the virus can enter into your system. But they are not in the upper respiratory tract for poultry virus specific viruses.
Have you found any low pathogenic H5 isolates in your
No LPA has.
no low pathogenic H5?
No, we have not got. Actually, the source is for us is the state. If state low pathogenic, it does not kill. When there is no problem, the state will not send this virus samples. And problem will come when there is high pathogenicity and when there is high mortality. So this is very straightforward. We don't go and collect from the from the mortality areas. We collect from the livestock markets and other places, but high mortality area we have not collected samples.
Any more questions?
Yeah, we are almost at the end of the technical session.
Any more
If
questions?
there are any more questions,
Open up the
questions, maybe we can have before we go to the session.
I have two questions.
Sir.
How many incidences of transmission from chicken to animals is reported in India?
Chicken to animals means which animal?
Chicken to humans.
Humans, yes. There are 5 incidents of human transmission. 2 are for H5N1 LPI and 3 for H5N1 viruses. 3 HPI viruses, one is in 2021 in Haryana, a child boy, I think age I don't know, he succumbed to the disease. The virus was isolated in 2021. Then in 2025, there is one again from this state, virus from this state, and again in 24, I think, from Andhra or Telangana, I am not sure, but 3 for H5N1, 2 for H9N2. H9N2 again in one in West Bengal and another in I'm forgetting the state, but 5 human transmission of avian influenza from poultry.
Were all the cases fatal?
No, for H9N2 they are not fatal, but all the H5N1 cases they are fatal. Yes.
And then second question is, say from March onwards, there's on a massive scale, vaccination against LPA is going on in India, right? From GP breeding stock, parent breeding stock and layer commercials, all of us have been doing now. Is there a surveillance happening from government of India or a state government side to find out how effective this vaccination is in preventing field incidences of LPA?
That is a good one, but actually there is a need. So far there is no surveillance in the vaccinated populations. Surveillance is already there, active surveillance, passive surveillance, but from the vaccinated populations there is no sample submission from the because one of my slides, the responsibility of the industry, the responsibility that they have to submit the samples to the RDDLs through the state departments or to the NIHSAD, because yes, as you are telling, there is no monitoring in the vaccinated populations, I can say. So, to my understanding, there is no monitoring of the vaccinated populations, but yes, surveillance is there. The routine surveillance of active and passive surveillance of Government of India for the poultry, but whether these samples are collected from the vaccinated population or not, we don't have that data.
I have a suggestion in this regard. If from the NIHSAD side, to whom all you have sold this technology, if you can write a letter to them or request them or make it mandatory, whichever the way you fit better, to submit data on this, certainly it will help in if at all any changes are required.
Yeah, yes.
In the vaccine seed, or will that can continue? That you will come to know only when you get these steps.
Yes, yes.
Unless
That is a good suggestion. Yes.
Unless that surveillance is taken up, because you have SRDBs taken and your lab is there central, and all the state governments are empowered to collect sample whenever they need. We from the private sector, we don't really move forward to give that, because it is like it is like today among general public, whether it is the farming community or a general public, our base mujhe maar, we do not want to do that, right? To understand it better, we do not want to move ahead and invite trouble, because we do not know if I start giving the data, how government will analyze it and what action it will take or what it will demand from me, I do not know. Hence, I on my own will not come forward to give any data on this. However, if you put a condition to those companies to which you have given the technology that you should give back this data on us, then they will be compelled to give that to you. The whole process, you get the data. The surveillance will happen automatically.
Yes, but the department should be in the loop actually for the central department and the state government also.
Sir,
Yes, your suggestion
my
is
suggestion is start at some point.
Yes.
See, let's say that there are so many Indian states. Not every state is a big poultry production farm. There are only some states which are having a very heavy poultry production in them. Okay, Karnataka is one of that. Tamil Nadu is one of that. AP, Telangana, Maharashtra, Haryana, West Bengal, these are all the heavily poultry producing states. In these states, if some initiatives are taken in these states through the state government, SRDDM, as insisted by you, some movement will happen. No movement which involves public can never be success overnight. It takes a long time. But my suggestion is please take an initiative.
Yes, that is a good one.
Please take an initiative. Otherwise, now we know that LPA vaccine is allowed, technology is sold, is given by you to all these vaccine companies, it is going on. I, as a farmer, can think, has it become another tool for this vaccine company to earn more revenue? You understood that? I do not know whether it's really working or not, or whether it is necessary or not. Please do some initiation, take up that. Call all these vaccine people to Bhopal, you ask them, we need no data. If you fail to give the data, if you refuse to give the data, then we need to review whether to give the technology or stop giving technology.
Yes.
Right? No one pays income tax on their own, right? Is there anyone? If tomorrow Government of India says paying income tax to Government of India is optional, how many of us pay? None, right? Because there is a compulsion, because there is a notice. Unless you ask, private people will not give because they are commercial people. Please take up that.
Yes. Thank you.
Study the impact of the vaccine.
My major objective is what is the impact of the vaccination? Is it really working? If not, what to do? Or if any further improvement is to be made, what is required? These are very important things.
Yes, that is a good idea. Yes.
So, if no more questions are there, then I request our Dean sir to propose vote of thanks. Please sit down, sir.
I will sit there.
Good evening, everybody. I think come to the end of the very exhaustive technical presentation. In fact, after almost a year, we have a very good presentation here. I remember last year, Dr. Bhatt from USA came and similar kind of interaction we had. It's my honor and privilege to propose the vote of thanks during this occasion. Dr. Chakradhar Tosh is also we are classmates. They are microbiology. I was at animal nutrition at IVR Izzat Nagar way back in 1989 to 1991. I thank Dr. Tosh for all the way coming from Bhopal, ICAR-NIHSAD, and upon accepting our invitation to make a presentation on this very, very important topic of the present day need. So a lot of technical information starting from basic information, the industry, research, so many issues were discussed, really a wonderful afternoon session. So I thank on behalf of the KVAFSU-KPFBA this center for giving a wonderful talk and making this technical session very, very fruitful. Now I request Sri Prasanna, Honorable Secretary of KPFBA, to come to the dais and present a memento to our speaker. I request Dr. Tosh to accept our memento on this occasion. Yeah, please. Yeah, please, sir. Prasanna, sir. Yeah.
ಸ್ವಲ್ಪ ಫ್ರಂಟ್ ರೈಟ್. Thank you, sir. You have come from a long way only for this purpose and enlighten us on this. Please take it.
You are always in the laboratory the 4 months of the laboratory. When you may talk also, I don't have much experience on the feed situations. Because you don't get, and people tell why don't you come to the feed during the outbreaks? Because but we cannot understand when the feed outbreaks are going on. We don't have any holidays or Sundays or Saturday or prayer lines. Because they are national emergencies, we have to do the events. So during that time, we intend to go to see how really these clinical symptoms and signs. But the laboratory concerns it's that there.
Yeah.
So that is the only thing. So I would have this opportunity to come back to what the need of the school and to investors. So thank you for this opportunity also.
Please take it.
This event is possible because of the constant support by the KPFBA. I would like to thank Sri Naveen Pushpati. Of course, he is not here. Of course, his constant support made this event possible. And Dr. H5N1 Shetty, of course, is always there with us, Chairman of the Technical Committee of KPFBA, and also Mr. Prasanna, General Secretary of KPFBA. We are thankful to you, sir. And also the Sajid and the staff of this KPFBA Center. We are thankful to you, sir. And we have the participants from Veterinary College, Dr. Islo, and he is the key person coordinated between the college and the KPFBA and Dr. Tosh for making this possible. Thank you, Dr. Islo and team. And also I thank the participants from HNDB and IVRA and members from industry and members of KPFBA and all the participants present here for listening to the talk and actively participated in the interactive session. So thank you so much.
Thank you.
Okay, thank you, sir. Thank you.
Okay, thank you.